Today I'm going to talk with you about the genetics underlying forensic DNA analysis. Forensic DNA analysis is a very powerful tool to identify the DNA of individuals. Unless you have an identical twin, you have a unique combination of genes in your DNA. So if you leave biological evidence at the scene of a crime, a forensic scientist can collect some of the blood and go back to the lab and analyze what combinations of genes are present in the sample. We call this a DNA profile. Once the forensic scientist has a profile, they perform a statistical analysis to determine who has the genes that match this evidence and calculate the likelihood that combination can be found by chance alone. DNA profiles can also be used in paternity tests to determine the likelihood that a child is descended from two parents. To understand this, I need to explain some simple genetics and define some terms. Genetics is the study of genes. Genes are discrete pieces of hereditary information called traits. For example, hair comes in straight, wavy, or curly. Your hair shape is inherited from your mother and father. The hair shape is coded for in a chemical called DNA, or deoxyribonucleic acid, which is decoded by the cell to make the proteins in your hair. Genes are lined up on thread-like things called chromosomes. So these are symbolically your chromosomes. The chromosomes come in pairs. All the cells in your body have two pairs of the chromosomes that are there. And there are thousands of genes on one chromosome. The long strand of DNA in the chromosome can be wound up in the nucleus by proteins, and within that long strand is a series of individual regions that code for the protein that determines the shape of your hair. Now I'm simplifying a bit since hair is complex and several genes are known to control its shape. But in humans, a cell nucleus contains a lot of chromosomes, 46 individual chromosomes or 23 pairs of chromosomes. Half of these chromosomes come from one parent and half come from the other parent. So if this is your father and this one came from your mother. And on these chromosomes are a series of different genes. So each parent can only give you one of their chromosomes, and which chromosome you received from your parents was inherited independently. So you're half of your mother's DNA and half of your father's DNA. So that means at least half of the DNA your mother had didn't come to you as well, and half of the DNA your father had didn't come to you as well. So if you think back to cell biology, you can recall that there's a process called meiosis, where a cell undergoes a reduction division. So in this case, you wind up with half the chromosomes in the sperm cells and half the chromosomes in the egg cells. So these will be our little sperm, and meiosis makes 4 sperm cells, and it makes 1 egg cell and 3 little polar bodies. So each one of these has half the chromosomes that your parents had. So I mentioned there were 23 pairs of chromosomes, so each one of those 23 pairs of chromosomes is divided in half. So now you've got 23 chromosomes in your sperm and 23 chromosomes in your egg, and when they reunite to make a fertilized egg, what you have is the full 46 chromosomes back together again. So the one thing that's different is that if you're a male, you are an XY, so you have a Y chromosome, and if you're a female, you have 2 X chromosomes. Otherwise, you have a group of pairs of chromosomes. So like the chromosomes, the genes come in pairs. So if this is symbolically our gene for hair shape, it's located along the DNA in one of the chromosomes. Now, it's possible that there's variation in this, so that instead of having just exactly the same DNA sequence, you can wind up with a different DNA sequence for some individuals. So this individual would have a slightly different version So maybe this is the person who has straight hair, and this is the person who has the curly hair. So you wind up then being a combination of the straight gene and the curly gene. So as I mentioned, each parent can only give you one of their chromosomes, and so each chromosome pair is inherited independently of the others. So you are a mixture of the chromosomes from your parental grandparents and maternal grandparents. And that explains why your brothers and sisters are not your exact twins. They've gotten a different group of chromosomes because each division of these inherits the chromosomes separately. Identical twins are a special case. They start out as one fertilized egg that came together from one sperm and one egg, and then they split into two individuals early in development. So identical twins share all the same chromosomes. So I've mentioned several times that the chromosomes contain DNA and then some structural proteins that are used for winding them up during cell division. So a DNA molecule is a repeating structure of 4 basic building blocks called deoxynucleotides. The 4 types of deoxynucleotides differ in which side chain they've got. And so the 4 types are adenine, thymine, guanine, and cytosine. And so we commonly refer to them as A, T, G, and C. So the DNA structure then is consisting of a double helix of 2 strands. So here you can see one strand of DNA, and here's the other strand of DNA. And one aspect of this is that the adenine, the A, always pairs with the T, and the guanine always pairs with the C. So each one of these steps in this kind of circular ladder that's twisting along consists of either an A-T pair or a C-G pair. And one strand then is the complement of the other strand. So you can take one strand, like if we look here, we can see here's a T, and so here's a C, and here's an A. So if we're looking at the other strand, we can predict if this is a T, this strand must be an A. And we can predict if this is a C, this one must be a G. And that's what we mean by the strands being complementary. So this way a DNA molecule can unwind into 2 separate strands and use each strand to make a copy of itself. So when one of them's copying this strand and this one's copying this strand. So the order in which the bases are arranged on the chromosomes is called the DNA sequence, and the complete DNA sequence of a human is called their genome. So there's a lot of biological information that has to be decoded by the cell in order to make proteins. And so a lot of the information that's in the DNA is there to make the proteins. But other information is only present as DNA. It never goes on to become a protein. And surprisingly, that's most of the DNA information. As much as 99% of your DNA never goes on to become decoded into a protein. So a lot of the traits then that are in this DNA are some repeated sequences of nucleotides. So A's, C's, G's, and T's that wind up repeated in a particular pattern. And these repeated DNA regions are what we use in forensic DNA analysis. These regions are chosen because they're biologically not as important as the protein coding regions. So if you find out information about these repeating regions, it doesn't really tell you anything about your risk of a genetic disease or any other genetic trait that you might have. And because they're not biologically critical to your survival, they can accumulate more genetic changes with no bad results. And so, as a result, they're quite variable. So forensic scientists have identified a group of these genes, a group of these regions in the genes, that they call short tandem repeats, or STRs. So we're going to call them STRs. So the STRs then are short sequences that are repeated of DNA letters one after another. So, for example, let's start with a simple STR repeat, which is ATTG. So this little repeat then, there's one unit. So if I'm making 3 repeats, I would just go ahead and make another one and another one, ATTG. And so these wind up being repeats, and this repeat has 3 repeats of the same little stretch of DNA of our basic unit of ATTG. And there are a lot of different potential numbers of these repeats that are possible.